Hi, everyone. My name is Eleanor Hansen, and I'm a research assistant at the Developmental Affective Neuroscience Lab at Columbia University. And today, I'm going to talk about our team's project, which was a review and meta-analysis of associations between early life adversity and children's brain structure using MRI. So, most neuroimaging research on children and early life adversity has been conducted with small sample sizes and a wide range of ages and adversities. Also, while many articles qualitatively review this research, there aren't yet many quantitative meta-analyses reviewing it. And further, nearly all existing meta-analyses focus on adult samples and self-reported adversity. So to build on and extend this previous work, our project sought to review and quantitatively summarize existing research on early life adversity and children's brain structure. To do so, we found our studies using an algorithm of search terms in several databases. To be eligible, studies needed human subjects ages birth to 18, at least one childhood psychosocial adversity, a measure of brain structure, sufficient and available data for our analyses, no significant comorbidity, and no duplicate samples. After screening, 130 articles were included in our qualitative review and 110 articles in our quantitative meta-analysis. Then, to analyze the data, we started with mixed effects models to look for main effects of adversity on different brain regions. For each region, we also tested for study heterogeneity, publication bias, and used leave one out analyses. We then looked at possible moderators, like age, gender, and adversity type. Specifically, one key adversity moderator we examined was interpersonal versus non-interpersonal adversity. So broadly, this means whether the adversity the child experienced directly affected their relationship with a caregiver, like maltreatment, or if it was more environmental in nature, like socioeconomic status. So, I can now share some of our results of the meta-analysis with you for the hippocampus, amygdala, anterior cingulate cortex, insula, ventromedial prefrontal cortex, and dorsal medial prefrontal cortex. And these regions are of particular interest because they are involved in things like emotional processing, learning, and memory, which we know are affected by adversity on a behavioral level. So for the hippocampus, there was a main effect of adversity, meaning that adversity was associated with smaller volumes across sample ages and adversity types. There was also a significant effect of adversity type, such that on average, interpersonal adversity was associated with smaller hippocampal volumes than non-interpersonal. And you can see these in our forest plots here, which show the correlations between adversity and brain volume for each study, and then for the total sample with the diamond there at the bottom. And you can see that the full range is a non-zero correlation. Then for the amygdala, there was no main effect of adversity. But however, there was a significant interaction between age and non-interpersonal adversity on amygdala volume. To look at that, I can orient everyone to this graph here on the left. Each dot represents a study, and the size of the dot is about how large its sample was, which affects how much weight it had in our model. The x-axis is the age of each sample when scanning occurred, and the y-axis is the effect of adversity on amygdala volume. Values above 0 mean that adversity was related to larger amygdala volumes, and values below 0 mean that adversity was related to smaller volumes. So here we can see that in childhood, adversity might be associated with smaller amygdala volumes, but in older samples, that might not be the case. Another thing to note is that our early childhood data here is pretty sparse. However, we did conduct leave one out analyses for each study with no significant changes in the results. On the right, it looked like interpersonal adversity might show the opposite effect, where increasing age is associated with smaller amygdalas. However, this effect did not reach significance with a p-value of 0.06. So next, we found a significant main effect of adversity on the anterior cingulate cortex, such that adversity was associated with smaller volumes. And additional analyses showed that these patterns were driven especially by the ventral anterior cingulate cortex. There was also a subgroup difference, such that samples with interpersonal adversity were more likely to have smaller volumes than non-interpersonal. And finally, there was another adversity type and age interaction. So here on the right, we can see a negative association of interpersonal adversity with age on anterior cingulate cortex volumes. And we can contrast that with the non-interpersonal adversity graph on the left, which doesn't show a clear pattern with age. Next, for the insula, there was a significant main effect of adversity, such that on average, adversity was associated with smaller insulas. Then for the ventromedial prefrontal cortex, we found a significant negative association of adversity on volumes. There was also another subgroup difference, such that non-interpersonal adversity was associated with smaller volumes than interpersonal. And lastly, there was another interaction with a negative association of overall adversity with age on ventromedial PFC volumes. And finally, for the dorsal medial prefrontal cortex, our data showed a significant negative association of adversity on dorsal medial PFC volumes. We also found another subgroup difference, such that non-interpersonal adversity was associated with smaller volumes than interpersonal. And there was another adversity type and age interaction, with a negative association of overall adversity with age on dorsal medial PFC volumes. So that's all the data I have to present today. In sum, our results can be organized into a few different kinds of effects. First, there were main effects of adversity, associated with reduced hippocampal, anterior cingulate cortex, insula, and prefrontal cortex volumes. There were also subgroup differences, such that interpersonal adversity was associated with smaller volumes than non-interpersonal for the hippocampus and anterior cingulate cortex. And conversely, non-interpersonal adversity with smaller volumes than interpersonal for the ventromedial and dorsal medial prefrontal cortices. And finally, there were multiple types of age and adversity interactions. Non-interpersonal adversity positively interacted with age on amygdala volume. Interpersonal adversity negatively interacted with age on anterior cingulate cortex volumes. And overall adversity negatively interacted with age on ventromedial and dorsal medial PFC volumes. So a couple important caveats of the interaction analyses are that, one, we use the mean sample age of each study, which does not capture the full range of the sample, and two is that we had relatively few studies in some cases. Overall, our review replicates past studies in adults that demonstrate an association between adversity exposure and decreased hippocampal and cortical brain volumes. Our findings also indicate many potential moderators of brain volume, including adversity type and sample age. So this further highlights the need for future research that focuses on specific aspects of children and their early environment to better understand how adversity gets under the skin to impact the developing brain. So I didn't have time to present all of our data, but I would love to share it with you. So please feel free to reach out to me via email or Twitter as listed here. I want to give a special shout out to my mentors and collaborators listed here, and thanks for listening.